Hey everyone, my name is Aditya Prabhakar, and today we're going to be going over specific types of channel inactivation. So I've made a previous video on specifically the ball and chain method of inactivation and activation, which you can find right up there. But today we're going to be talking about fast inactivation, slow inactivation, calcium-dependent inactivation. Before I get into that, let's talk a little bit about how channels are activated. So voltage-gated channels become activated when they are introduced to a sort of depolarizing current. And this can happen through methods such as the sodium-potassium pump that initially open during depolarization, and after they open, they quickly transition into an inactivated state where they're temporarily unable to open again, even if the stimulus is present. And after this, they'll return back to the resting state only when the membrane potential also returns back to its resting levels. So let's go straight into what types of different inactivations there are. We're going to start off with fast inactivation. So with fast inactivation, we normally associate this with the Hodgkin and Huxley model. So Hodgkin and Huxley model. So a really prime example for this is your voltage-gated sodium channels. When the membrane depolarizes, sodium channels open briefly, allowing sodium ions to rush inside your cell. So they're coming from the outside, from the extracellular side, and flowing inwards because of the concentration gradient into the intracellular side. So after this, these channels become inactivated, even if the membrane is depolarized. The mechanism of how this works is there exists a blocking particle, and like I said before, it's similar to the ball and chain method. Like I said, that video is linked right up there, so go ahead and watch that if you need a little bit more background on that. But this ball basically just plugs in your actual port of transportation. So where this sodium goes in, this eventually gets blocked out by a separate ball method that goes and fits inside there. So this generally occurs after the channel is open and prevents any further sodium ions to flow into the cell, and the inactivation process itself is removed only when the membrane is repolarized and allows the channel to go back to its closed state and it's ready to open again for the next activation. So the benefit of this function is that fast inactivation, it's very critical for the unidirectional propagation of action potential, so it's one way, and it ensures that the sodium channels don't stay open for too long, which would disrupt the ability for any neuron or cell in general to reset for the next signal. But specifically for this purpose, we're going to talk about neurons. Next, let's talk a little bit about slow inactivation. So the prime example for this is your voltage-gated potassium pumps. The way this happens is it happens on a very similar method to our sodium pumps, but on a slower time scale. So it's more of a subtle conformational change. The same ball and chain method is going to be utilized. So it involves the actual structural rearrangement of the channel's pores to block the ion channel itself. So when potassium starts to flow into the cell and it reaches your depolarization, your channel stops and it's a lot slower, and the change occurs over a greater scale of the actual cell. So the function of this is that slow inactivation helps regulate prolonged electrical signals and ensures that the neuron doesn't remain hyperpolarized for too long. And the last method I'm going to be talking about are our calcium-dependent inactivation channels. So these are our voltage-gated calcium channels, and they respond to the high accumulation of calcium within the cell after the calcium channels open. And the higher internal concentrations bind to regulatory proteins such as calmodulin, which you'll see a lot in this class, which leads to the inactivation. So the way this works is just like the other models, to prevent excess calcium from flowing into the cell, because excess of all these molecules can be toxic to the cells. It allows it to stop and act as like a regulatory function until the cell goes back to being at its resting states. So to go over the two bigger topics that we talked about today with the first two methods, it's the ball and chain method, which occurs for the sodium channels, and the similar method, but the structural change method for our potassium channels. So that's the two biggest separators. The sodium channels, ball and chain, potassium channels, structural changes. So again, sodium channels, ball and chain, potassium channels, structural changes because the actual pore itself is being changed. So an example of how this might be relevant to us in the real world is it's linked to a lot of diseases when you don't have the channel inactivation. So for example, you have long QT syndrome, which occurs when you have mutations in potassium channels. So the slow inactivation doesn't work, which actually blocks your pain signal so you can't feel pain, which inherently doesn't sound like an issue, but when you concur in a very severe injury, you're going to want to feel that pain, so you know how to like react to it with your other systems. So yeah, the points that you should emphasize or get from this video are the fact that we have different types of channel states. So we have our depolarizing state, resting state, open, inactivated, and they're all correlated to some sort of channel function. The recovery from inactivation. So even after you reach that peak of being really activated and you go back down to being inactivated, it takes a moment for you to go back to that resting potential where all the channels are now closed but can be opened again. Remember, during this inactivation period, nothing can happen. And the inactivation timing, so that's talking about our fast and our slow and the differences between those two. Awesome. I hope you found this video a little bit helpful. If you need any other additional information or any written graphs, I'd be more than happy to do that as well. But happy studying and good luck on your first test.